Hello everyone, this is Orkumita Talukdar. Welcome to my final year thesis presentation. My project is multimodal data fusion and machine learning for deciphering protein-protein interaction. It is a research project and my sponsor is Dr. Yang Shen, Assistant Professor, Electrical Engineering Department, Texas A&M University. This would be the outline of my project and my presentation. I would explain what are proteins and their relevance, the needs and objectives, current methods and our innovation, the approach we are taking, performance evaluation, and lastly, conclusion and future direction. After spending a significant time trained to be an electrical engineer, I can relate the theories and the disciplines of electrical engineering everywhere, including our human body. If we think how human body works, it is actually very, very similar to an electrical system. Like an electrical system, we also have hierarchy. If we consider our body as device, the organs and the cells, tissues can be considered as the modules of those devices, like DAC or ADC, instrumental amplifier. These organs and tissues are enabled by three major biomolecules, DNA, RNA, and protein, very similar to resistors, capacitors, and inductors. Proteins can be considered as one of the most important biomolecules. It is also called the signal carrier in any physiology. We have proteins everywhere, in our eyes, in our skin, our nervous system, muscle development, blood vessel, everywhere. We all know about insulin. We need it to process our glucose. It is a protein. We also know about hemoglobin. It helps us to carry oxygen throughout the body. It is also a protein. Given the important roles protein play in our physiology, we must know what are proteins and how they work if we want to know how biological signal processing works. So what are proteins? Protein has a very simple primary structure. It is a sequence of amino acids bonded by peptide bonds. We have 20 different amino acids present in nature. We can think protein as a sentence written by the alphabet of these 20 basic amino acids. Other than this primary structure, it also folds as a three-dimensional form in human body, which enables protein to perform any functionality. Now, if we think about it, the capacitors or the inductors, they do not perform by themselves. They are connected to a wire, then they can perform any functionality. But human body does not have any wire. Rather, it has interactions. Very similar to those electrical modules or electrical units, proteins also interact with other proteins to perform any functionality. And that is the purpose of this project. We are trying to study how proteins interact in three-dimensional space. The needs and objectives of our research. Thanks to the advancement of biological science, we know a lot about proteins, but yet we know very little how they interact with each other. Here is a figure of hemoglobin given, interacting with 4 different chains, forming oxy and deoxy form. The current pandemic, coronavirus, it also uses protein interaction. In the beginning of this pandemic, doctors were very confused that why the human lungs are being so affected, why not heart or any other organ? That's because coronavirus has this spike protein and it docks with the ACE2 protein, which is present in lungs. That is why it affects lungs to dock with this ACE2 protein, thus infect our human body. So why solve this problem? If we can understand how proteins interact in three-dimensional space, we can study how drugs work in our body more effectively. A lot of diseases can be early detected using biomarkers. Given that every human being is very unique in his or her genetic formation, personalized medication can be developed. Lastly, any faster development of vaccine is possible, which was a necessity a couple of months ago. In fact, one of the vaccine mechanisms actually use this protein interaction. Using the vaccine, it just blocks this ACE2 bonding part so that coronavirus cannot come and dock with it. Thus, our human body can be protected. Current methods and our innovation. Currently, we have a lot of experimental methods like affinity purification, ECEN hybrid, and so on. But the problem with the experimental methods are they are very expensive, very time consuming, and given that we have so many protein interactions in our body, we cannot possibly figure out every interaction experimentally. We also have computational methods like protein docking, but the problem with the computational method is we have very less available protein structure data. That is why, given we have these methods available, we still have a significant gap predicting protein interactions in three-dimensional space. There comes the novelty of my project. Using the latest techniques of signal processing and artificial intelligence, we can model protein as a multimodal data. If we recall, we can represent protein as a sentence written by the alphabet of amino acids. Thus, we can represent protein as text. Protein interacts with other protein, even within itself. That interaction can be represented as a graph. Lastly, the three-dimensional formation of the protein can be represented as a 3D image. So here comes our innovation. Using that multimodal data, we are developing a data-driven machine learning approach to predict protein-protein interaction. We can also think it as a biological signal processing using machine learning. We are using text, 1D modality, and two types of graphs, 2D modality. For text, we mean that we are using sequence of proteins and we are using both positive and negative interactions. For example, here is a sequence of protein A, here is a sequence of protein B. 1 means they are interacting, 0 means they are not interacting. We're also using inter-protein residue-residue contact or inter-RRC map. When protein A interacts with protein B, this physical interactive bond can be represented using a graph. For example, in this residue, they are not interacting. That is why it is 0. In this residue, they are interacting. That is why it is 1. We're also using intra-RRC map, which demonstrates the physical interactive bond within that protein. We're using this 1D modality and 2D modality to train our model. So what we're trying to predict actually, what would be the output of the model? Number one, we're trying to predict the consequence, whether two proteins interact or not. We also call it PPI, protein-protein interaction. For example, given protein C and D, is it a 0 or 1? Number two, we're also trying to predict the cause or how two proteins interact. We call it inter-RRC or inter-residue-residue contact map. Given protein C and D, which are the residue positions, we have these physical interactive bonds and where we do not. The positive part of our approach is it is inexpensive, less time consuming. It can be applicable to a larger set of data, and we are overcoming the protein structure data limitation by using this multimodal data fusion. Approach and data curation. Given that it is a data-driven project, the data curation is very, very important. I would try to portray the entire protein interaction picture over here. Actually, we have a lot of positive interaction data available or positive consequences. We have almost 300,000 pairs of protein which are known to interacting with each other. On the other hand, we have only 6,000 pairs of protein which are known not interacting with each other or negative consequences. As I mentioned, we have a very small amount of structural data available. Out of these 300,000 positive interactions, we only have 2,000 structured data available. Removing all the redundancy and biases, I have chosen 5,000 positive interaction and 5,000 negative interaction, and I had 57 structural data available. Lastly, I have divided this data set as 80% training, 10% validation, and rest 10% as test set. Model development. Model 1 takes 1D modality or the sequence of proteins as input. The label is PPI. We're using HRNN or hierarchical recurrent neural network to embed this 1D modality or the sequences. The HRNN is one of the latest machine learning techniques, which is largely used in NLP, natural language processing, or text processing. Using a naive attention, we are trying to predict both PPI and inter-RRC map. The loss is described as balanced cross entropy, BCE loss for PPI, because our label is PPI. Model training algorithm, ADAM. For training environment, we have used ANM high performance research computing facilities, cluster Terra. The novelty or the exceptionality of model 1 is we are only using the big data of consequences, but trying to predict both consequences and causes. So one point to notice here, though we're trying to predict these causes or inter-RRC maps, we have not given any inter-RRC data to train this model. That is why this attention is very naive. This model still does not know where to put the attention. But if we recall, we have a very small amount of data of causes available, which was 57 inter-residue-residue contact map. So is there a way to combine this big data of consequences and small data of causes? There comes model 2. For model 2, we are also using 1D modality as input. Now the label has changed. As label, we are using inter-RRC map, this 57 inter-RRC map. We're using HRNN to embed the 1D modality. Now we are using a smarter attention mechanism, and we are trying to predict the inter-RRC map. For loss, it has been changed because our label has been changed. Now it is BCE loss for RRC. Now how the model 2 combines this big data and small data? The embedding part of the model 2 has been initialized by the optimized weight we got from model 1. This is also called warm start. We expect this warm start in model 2 somehow imprints the memory of big data which was used in model 1. Later, using this label, inter-RRC map label, the model 2 is being trained and fine-tuned. Using this warm start and fine-tuning, this attention got smarter. Now it knows where to put the attention. If we recall, other than the 1D modality, we also have 2D modality structure available for proteins, which was intra-RRC map. In model 3, we have fused these two modalities. As input, now we are taking both 1D modality sequence and 2D modality intra-RRC map. The label is inter-RRC map. We are using HRNN to embed the sequence. GAT or graph attention network is another very latest technique of machine learning, has been used to embed the graph. Now the attention is fused because two different modalities has been fused, and the output is inter-RRC map, or that is what we are trying to predict. Performance evaluation. While we are trying to predict the consequences, whether two proteins interact or not, or PPI, the model 1 performance has been compared with one of the current methods. The name of the paper is PPI Detect. It is a vector machine-based paper. In this case, model 1 has been trained in their dataset, and we see when we use Gmin to as threshold to binarize our prediction, our precision goes up as 7.7%, the sensitivity goes up as 9.57%, and the accuracy goes up as 6.2%. Model 1 has been also trained with our curated dataset, and we have used AUPRC, area under precision recall curve, as the performance evaluation metric. We have used 5,000 positive dataset and 5,000 negative interaction dataset. That is why the random is 0.5 for both training, validation, and test. And we see that AUPRC beats the random value in all three cases. While predicting the causes, how proteins interact with each other, or the inter-RRC map, model 1, 2, and 3 has been compared with the random. Randomly, we have one positive residue-residue contact out of 10,000 contacts. Model 2 performs 9 times better than random. Model 1 performs 9 times better than random. Model 2 performs almost 22 times better than random. And model 3 performs 24 times better than random. Conclusion and future direction. Summarizing the entire project, we can conclude that when we use the big data of consequences, in case of PPI prediction, our PPI prediction beats the current state of the art. To predict the inter-RRC map, we the model 1, or we can perform 9 times better than the random. When we combine the big data of consequences and small data of causes, the model performs almost 22 times better than the random. Lastly, when we fuse both 1D modality and 2D modality, we have the best performance, which is 24 times better than the random. This project has a lot of future dimensions. This project can be extended to predict the complete atomic resolution. Alongside with the residue-residue contact, we are planning to predict the continuous angle, distance, overall the entire geometry of protein molecules. Lastly, we hope that our work, our project helps us to understand how proteins interact in three-dimensional space much better. Thank you very much. That was from all my side. You are most welcome to ask one question.